morning, and welcome to the Albireo Pharma Business Update Call. At this time, all participants are in a listen-only mode. A question and answer session will follow the formal presentation. If anyone should require operator assistance during the call, please press star 0 on your telephone keypad. Please note that this conference is being recorded. I will now turn the call over to your host, Hans Vitzthum, Managing Director of LifeSci Advisors. Thank you. You may begin.
Operator, and good morning, everyone, and thank you for joining today's call. This morning, Albireo issued a press release highlighting top-line results for the PFIC-1 Phase 3 pivotal trial evaluating Odevixibat in PFIC. This press release and the data presentation slides are accessible via the company's website at www.albireopharma.com. Before proceeding, we would like to note that management's comments today may include forward-looking statements regarding the company's plans and expectations. These statements are made under the Private Securities Litigation Reform Act of 1995, and they are subject to various risks and uncertainties. Actual results may differ materially due to various important factors, including those described in the Risk Factors section of our most recent Form 10-K and our subsequent SEC filings. These filings can be accessed from the Media and Investor section of our website at www.albireopharma.com or on the SEC's website. Any forward-looking statements represent our views as of today, September 8, 2020, and should not be relied upon as representing our views as of any subsequent dates. We undertake no obligation to publicly update these statements. Now, it is my pleasure to turn the call over to Ron Cooper, Albireo's President and Chief Executive Officer. Ron?
Thank you, Hans, and thank you everyone for joining us this morning. With me today are Simon Harford, our Chief Financial Officer, Dr. Pat Horn, our Chief Medical Officer, and Dr. Richard Thompson, Professor of Molecular Hepatology at King's College London and Principal Investigator for our PFIC clinical trial. We're fortunate to have him join us today to provide his medical perspective. Now to the news. We're excited to announce positive top-line results from the PFIC-1 Phase 3 clinical trial of Odevixibat, our highly potent non-systemic ileal bile acid transporter, or IBAT inhibitor, for the treatment of children with progressive familial intrahepatic cholestasis, or PFIC types 1 and 2. It is estimated that there are 8,000 to 10,000 children without an approved drug for this life-threatening disease. PFIC-1 is the first and the largest global, pivotal, prospective, randomized, placebo-controlled trial in PFIC types 1 and 2, which was designed to support approval of Odevixibat in multiple regions around the world. The study met its primary endpoints with a very high level of statistical significance for both pruritus and serum acids in both PFIC-1 and PFIC-2 patients. Odevixibat was well tolerated with a single-digit diarrhea rate consistent with our Phase 2 trial. So what did we see specifically? Statistically significant improvement in pruritus assessment, P equals 0.004. Statistical significant reduction in serum bile acid response, P equals 0.003. Both Odevixibat doses statistically significant for both endpoints. Similar efficacy in PFIC-1 and PFIC-2 patients. Excellent tolerability profile and a low rate of diarrhea or frequent bowel movements versus placebo. So why are these results exciting for us, children, families, and physicians? These results add to our body of data showing highly potent Odevixibat reduces serum bile acids and eases the pruritus, which is devastating for children and families living with PFIC. We're very pleased that our once-daily dose, taken by capsule or sprinkled over food without the need for refrigeration, demonstrated positive results in children with PFIC-2 as well as those with PFIC-1. These results reinforce the potential for a paradigm shift in the treatment of PFIC and represent a major commercial opportunity around the world. At the same time, the positive results from the PFIC-1 trial increases our confidence in our ongoing Odevixibat program for biliary atresia, our planned program in Alagille syndrome, as well as our preclinical bile acid modulator compounds for adult liver diseases. The results in PFIC are the first step in building Albireo's leadership position in pediatric cholestasis around the world, and we hope to help the estimated 30,000 to 40,000 patients with these terrible diseases. Odevixibat has received orphan drug designations in the U.S. and Europe, as well as rare pediatric disease designation for the treatment of PFIC with the associated priority review voucher eligibility in the U.S. We are thrilled about the opportunity to address this major unmet need globally, and we're now preparing submissions for the approval in the U.S. and Europe and advancing our commercialization preparations. I'll discuss these topics in more detail a little bit later. So to take you through data, I will turn it over to our Chief Medical Officer, Dr. Pat Horn, who will go through the study design and top-line results. Pat?
Thank you, Ron, and good morning. We are pleased with the positive results we observed in the PFIC-1 trial and happy to share the data this morning. I will review the results while referencing the presentation, which is posted on our corporate website. Before I do that, I'd like to emphasize the rigorous process behind the design of PFIC-1 and PFIC-2. In our early phase 1 study, we first observed Odevixibat demonstrating consistent bile acid reduction and a low rate of diarrhea in the lower doses. After consulting with regulatory authorities, we moved to the phase 2 study, where we observed PFIC patients eligible for the phase 3 study achieved a mean reduction in serum bile acids of over 70% in 4 weeks, a decrease similar to that observed in the bile acid diversion surgeries. We also observed a reduction in pruritus and an improvement in sleep, with no diarrhea observed during the 4-week treatment period. These results allowed us to move on to the phase 3, with eligibility criteria targeted to the patient population for whom efficacy was demonstrated in the phase 2 study, and a randomized double-blind placebo-controlled global multi-center clinical trial design, which is considered the gold standard. We evaluated Odevixibat in 62 patients ages 6 months to 16 years with PFIC type 1 or type 2. Patients were randomized to receive either a 40 microgram per kilogram per day or a 120 microgram per kilogram day oral dose of the planned commercial formulation or placebo, and they received these once daily for 24 weeks. If you refer to the slide deck we posted this morning, I'll be walking through slides 6 through 11. On slide 6, this is the primary analysis. The study met the U.S. regulatory primary endpoint, with the proportion of positive pruritus assessments being 53.5% in the Odevixibat arms compared to 28.7% in the placebo arm, yielding a p-value of 0.004. Pruritus was measured every morning and every evening. The proportion of positive pruritus assessments were measured by comparing each of these measures versus the baseline. If the measure was 1 point greater drop, it was deemed a positive assessment. On slide 7, you can see the change in absolute pruritus measures on a 0 to 4 scale. Outside expert analysis has determined that a 1 point or greater reduction is a clinically meaningful reduction. Patients on Odevixibat had a 1.13 mean decrease from baseline versus a 0.25 for the placebo group, and this was statistically significant with a p-value of 0.02. Slide 8 shows that the study also met the EU regulatory primary endpoint. The definition for success in the bile acid reduction endpoint was either a 70% reduction in serum bile acids or reaching a serum bile acid level of 70 micromole per liter or less. No patients in the placebo arm met this definition. 33.3% of the patients in the Odevixibat arm did meet this definition with a highly significant p-value of 0.003. On slide 9, in a secondary analysis, placebo patients had an absolute mean increase of serum bile acids of 13.1 micromole per liter over the 24 weeks. On the other hand, patients on Odevixibat had an absolute mean reduction of serum bile acids of 114.3. Again, highly significant with a p-value of 0.002. On slide 10, you see the data for the split of PFIC-1 and PFIC-2 patients. In this study, 17 of the 62 patients, or 27%, had PFIC-1. The proportion of positive pruritus assessments and the reduction of bile acids were similar in PFIC-1 and PFIC-2 patients. Odevixibat was well tolerated as demonstrated on slide 11. There was minimal difference between the placebo and treated group for overall treatment emergent adverse events, and there were no drug-related serious adverse events reported on this study. On slide 12, drug-related treatment emergent adverse events were 3 or 15% in the placebo group and 14 or 33.3% in the Odevixibat group. As you would expect in these young children, the most common adverse event was infections, such as ear infection, upper respiratory tract infection. Diarrhea or frequent bowel movements were the most common treatment-related gastrointestinal adverse event, and this occurred in 9.5% of Odevixibat-treated patients versus 5% of placebo patients. Given that the mechanism of IBAT inhibition is to divert bile acids into the colon, this rate of diarrhea is very low, considering the significant reduction in serum bile acids. As you can see, we have demonstrated efficacy and tolerability results for Odevixibat in both the phase 2 study and now this phase 3 study. We will continue to learn more about the benefits of Odevixibat as we continue to analyze the data from the PFIC program, which is the largest data set ever created evaluating IBAT inhibition in PFIC patients. It's important to note that beyond PFIC-1 and the top-line results we are reviewing today, the full data set includes PFIC-2 study results as well. PFIC-2 is our open-label extension study, which is designed to assess long-term safety and durability of the response to Odevixibat. Cohort 1 of the trial allowed patients from the PFIC-1 study, the study we're discussing now, to continue treatment with Odevixibat. A second cohort, cohort 2, consists of patients who did not participate in the PFIC-1 study and includes other forms of PFIC not included in PFIC-1. We will continue to analyze this large and robust data set with the intent to present our findings at a future scientific meeting. Now I'll turn it back to Ron.
Thank you, Pat, and thank you and your team for designing, executing, and analyzing an excellent study. We appreciate it. It's my pleasure to introduce Dr. Richard Thompson, who is the principal investigator for the PFIC-1 and 2 clinical trials. Dr. Thompson is a professor of molecular hepatology at King's College London, an honorary consultant pediatric hepatologist at King's College Hospital in London. He specializes in pediatric liver disease and is particularly interested in genetic liver disease in both children and adults. Through worldwide collaborations, Dr. Thompson's lab continues to identify new causes of genetic liver disease. He served as the clinical lead for genomics and the 100,000 Genome Project at King's College Hospital, and is the clinical lead for a diagnostic laboratory specializing in liver and gastrointestinal disease. Before turning the call to Dr. Thompson, on behalf of Albireo, I'd like to express our appreciation for the many years of excellent collaboration and for leading this important study. Dr. Thompson?
Thank you, Ron. I do really appreciate the opportunity to share my perspective on what these data mean for clinicians and patients and for the future treatment paradigms of cholestatic liver disease. PFIC, or progressive familial intrahepatic cholestasis, is a rare genetic disorder that causes progressive and life-threatening liver disease. Children diagnosed with PFIC have impaired bile flow or cholestasis, caused ultimately by underlying genetic mutations. The resulting bile acids build up in the liver and cause the cells damage and the liver disease and symptoms. And the most prominent and problematic ongoing manifestation of the disease is pruritus, or intense itching, which frequently results in a severely diminished quality of life. Patients often also have jaundice and poor growth. We currently don't have any drug therapies available to use for these patients beyond off-label treatments, and we have frequently reverted to surgical options, and in particular partial external biliary diversion, or PEBD, and very often then liver transplantation. Of course, both of these interventions carry their own substantial risks. So therefore, it is a significant advance to have data on these 62 patients, the largest study of PFIC to date, demonstrating a statistically significant effect on both serum bile acids and an improvement in pruritus in patients with PFIC. That, coupled with the favorable safety and tolerability profile that Odevixibat exhibited, these data underscore the potential that IBAT inhibition and Odevixibat could truly have in transforming current treatment protocols and shifting the treatment paradigm to providing PFIC patients with a new drug option in the case of Odevixibat. So the reductions in pruritus that we have seen in this study have been shown to equate to real improvements in quality of life, and the reductions in serum bile acids that we have seen, we have shown recently in a surgical study of interruption of the intrahepatic recirculation of bile acids to correlate with very significant reductions in the need for liver transplantation. So I really want to take this opportunity to express my appreciation for all my fellow investigators, and obviously in particular the patients and families who have participated in this study. Without their enthusiastic participation and commitment, we could not have completed the trial at all, never mind on time. So thank you, and I'll pass back to Ron.
Thanks, Dr. Thompson. Our mission as a company is to provide hope to families. I'd like to amplify your remarks, Dr. Thompson, by saying that healthcare providers give hope to these PFIC families on a daily basis. And we're really glad to be able to share the results and milestones with the investigators and the entire liver community. So where do we go from here? Our focus now is on regulatory filings, commercial readiness, and advancing the Albireo pipeline. The regulatory effort will be led by Martha Muffy-Carter, our Chief Regulatory Officer, and Dr. Pat Horn, who between them have already successfully completed 10 NDA BLA filings. That is correct, 10 filings. Albireo plans to complete regulatory filings with the FDA and EMA early next year. We've been preparing for these regulatory filings for over a year, and draft versions of modules 1 to 5 are in place. We can now populate these modules with the phase 3 data. Following completed regulatory submissions, we anticipate approval, issuance of a rare pediatric disease priority review voucher, and launch of Odevixibat in the second half of 2021. Our organization has considerable, successful commercial experience to execute a successful launch. Pamela Stevenson leads our commercial organization and has a decade of Pfizer experience and another decade helping build Vertex and launch rare products. In addition to Pamela, both Simon and I have significant global launch experience from our years at Lilly, GSK, and BMS. Our new head of the Americas and new head of international have already been secured and will join us soon following this positive data announcement. They will be responsible for building commercial organizations in each geography. We've continued to build out our medical affairs teams with leaders and MSLs joining us across the United States and Europe. For the U.S. market, we've been refining the number of prescriber targets that will define our call universe through extensive market research, account mapping, and other analysis. Based on our learnings, we plan to hire a field force of about 10 people to cover the prescribing base. We've also conducted extensive market research with payers who expressed interest in learning more about PFIC, recognize the high unmet medical need, and would like to see the data. With the positive phase 3 data, we can now populate the value dossier and key economic models. Our plan is to have a compelling pricing and reimbursement submission to maximize access. For commercial supply, we're in the process of developing specialty pharmacy network model, and we are full steam ahead with manufacturing and supply chain plans as we continue to hit key milestones. Overall, we are ahead of schedule with our key organization, physician, payer, and supply milestones as we plan for the launch of Odevixibat for the treatment of patients with PFIC in both the U.S. and EU. Let me now move to speaking about advancing the Albireo pipeline. So with today's announcement, Albireo has now brought 2 different drugs through phase 3 trials, and we're even more confident about our ability to expand the indications for Odevixibat. We also have a pipeline of compounds from our platform that will deepen and extend our reach. We look forward to telling you more about that as the year progresses. Beyond PFIC, our biliary atresia phase 3 study, BOLD, continues to progress nicely despite the challenges of COVID-19 with more site openings and patient enrollment. As a reminder, BOLD is a global precedent-setting trial in a disease which is the #1 cause of pediatric liver transplants around the world and the largest pediatric cholestatic liver disease. We estimate about 15,000 to 20,000 children suffer from biliary atresia in the U.S. and EU. Our third Odevixibat pivotal program will be in Alagille syndrome, which we plan to initiate by the end of the year. We estimate about 3,000 to 5,000 children suffer from Alagille syndrome in the U.S. and EU. Our investigational new drug application for Odevixibat in Alagille syndrome has been cleared by the FDA. As with the PFIC trial, these pivotal programs are gold standard designs and are intended to generate efficacy and safety data to support approval in multiple geographies around the world. Let me close by saying that I am proud that we have delivered the Odevixibat phase 3 study within guidance and according to plan. We believe that our once-daily, non-refrigerated, highly potent, and non-systemic bile acid lowering product has the potential to be best in class. Our multi-region leadership approach in pediatric cholestasis across multiple indications sets us up for strong commercial launch and a planned profitable business. This management team has demonstrated an ability to deliver on time, and I'm confident that we can do the same going forward. Overall, we are very proud of what we've accomplished as we continue to demonstrate our ability to drive programs forward toward achieving our vision of becoming a fully integrated biotechnology company with an active commercial organization and a pipeline of therapies under development. We thank everybody for joining us and are pleased to open the call now for Q&A. Operator?
Thank you. We will now be conducting a question and answer session. If you would like to ask a question, please press star 1 on your telephone keypad. A confirmation tone will indicate you are in the question queue. You may press star 2 to remove yourself from the question queue. For participants using speaker equipment, it may be necessary to pick up your handset before pressing the star keys. One moment, please, while we poll for your questions. Our first questions come from the line of Ritu Baral of Cowen. Please proceed with your questions.
Hey guys, good morning and congratulations. It's a really solid set and I'm really happy for you guys and for patients as well. Can you talk a little bit about any potential dose response? Just given both, you said both treatment groups were statistically significant. Did you see a dose response? What does that mean for your filing strategy? And can you comment at all about the time course of response as well as any sort of differential response for PFIC-1 versus PFIC-2 patients?
Yeah, Ritu, so this is Pat. So those are all excellent questions, and as you can imagine, we're actively investigating all of these. Right now, we're just really presenting the top-line data. We really just kind of looked at the primary endpoint, as was mentioned, that, both doses achieved statistical significance at both of the primary endpoints, and we're continuing to explore, maybe look into secondary endpoints to see if we can differentiate on that. Again, PFIC-1 and PFIC-2, the endpoints look solid for that as well, but, again, as you can imagine, as we continue to analyze this data, we'll formulate our plan and our dose recommendation moving forward.
Got it. Let me sneak another one in there then. And this is actually for Dr. Thompson. Dr. Thompson, as we look at slide 8 and the overall serum bile acid reduction of, the responder serum bile acid reduction of 33% of patients versus none. And as you look at absolute reduction means, what do you think this actually means for time to transplant benefit? Is there enough in the literature that you can say it delays liver transplant by X or has X disease modifying benefit?
Thank you. I think that's a really important question. Obviously, I'm obviously delighted that we seem to have changed the biology of the disease. We've improved the pruritus, but as you say, really at the end of the day, I want to keep these patients away from the transplant surgeons. And so obviously this study can't determine that in itself at this stage, but I think you're probably aware that the NAPID consortium, which is an independent academic consortium, we've been collecting data over the last 3 years, and we recently published the results of surgical diversion, and in particular, whether a reduction in serum bile acids after surgical interruption of the enterohepatic recirculation correlates with long-term outcomes. And in what we showed in that study is if in the first year after surgical intervention, if you drop your serum bile acids below 100 micromoles or 75% reduction from baseline, then that dramatically changed the requirement for liver transplantation with 15-year follow-up. In fact, if you achieve those endpoints, then that completely removed the need for liver transplantation in that group. So obviously the endpoint here, the 70% reduction or a greater than 70 micromole reduction was designed before those data are available, but they are, look on the surface at least, highly comparable, and we'll obviously reanalyze those in a fashion consistent with the NAPID data, but it looks extremely similar. So it looks like there's a significant proportion, it remains to be seen, but who have met those criteria and on the basis of those surgical data would correlate with long-term native liver survival. So I'm optimistic that there's a significant proportion of patients here who will have been saved from liver transplantation.
Great. And then just one follow-up and I'll get back in the queue. Can you guys talk about what the Odevixibat related SAEs were, and can you talk qualitatively about the diarrhea, the grades of diarrhea, severity of diarrhea seen in those patients?
Yeah. So again, this is Pat. So there were no Odevixibat related SAEs. There were no drug-related SAEs within the study. The treatment emergent, as we said, the majority of those, the drug-related treatment emergent adverse events fell into the gastrointestinal SOC and also in the investigation SOC. And there were some variations in bilirubin and ALTs, and that was observed in both the Odevixibat arm and the placebo arm, and really is representative of the underlying liver disease.
Thank you, Ritu.
Great. Thanks so much.
Thank you. Our next questions are from the line of Tim Lugo of William Blair. Please proceed with your questions.
Thanks for the question, and congratulations to the team on the strong data this morning. Especially given the COVID pandemic, it's very impressive how everyone executed. Maybe a question for Dr. Thompson. Just to kind of reiterate what you mentioned about the 100 micromole in the NAPET study, could you just, given the strength of data this morning, could you just tell us where this will fall versus PEBD surgery now? Is it safe to say that this is strongly something that should be considered for most patients before PEBD surgery?
The simple answer is yes. I think there's several aspects to that. First of all, PEBD surgery, obviously, I think has been underexploited, largely because it is a form of surgery and there's a considerable hurdle to using it. So I think there's lots of patients that could have benefited from surgery previously who didn't. But clearly, if it's a once-a-day non-absorbed drug, then the hurdle is going to be dramatically different. So I think these results look very comparable to surgery, but I think it will be accessible to more patients because of that lack of a reluctance in some families to undergo surgery. They will be much more inclined to try a drug, and I think I think we'll use it in the same way, and I think it's a genuine alternative.
That's great to hear. Thank you for that. And could you maybe for the whole team or Dr. Horn, can you talk about the treatment adverse event which led to discontinuation, just what occurred with that patient and some of the outcomes?
So that was a patient who developed diarrhea.
Okay.
So that was one
right.
patient. Thank you for the kind words, Tim. Appreciate it.
Thank you, and congratulations.
Thank you. Our next question has come from the line of Yasmeen Rahimi of Piper Sandler. Please proceed with your question.
Hi, team. Congratulations on the amazing data, and I'm incredibly happy for you. Two questions, one for you, Dr. Horn, and one for Dr. Thompson. So maybe can you shed light on how many patients in PFIC-1 rolled over into PFIC-2, and how important is that extension study in terms of NDA filing, and when should we be seeing data? And then the question for Dr. Thompson is, can you kindly help us understand the significance of a 1-point improvement in pruritus? What does that mean for patients and caregivers, and how much hope does it give to your patients that you have been treating for many years? And thank you again for taking our questions.
So Yasmeen, I'm not sure I remember the questions in the order. In terms of the rollover, well over 90% of all patients who participated in this study rolled over into the open label extension study. And I haven't looked at the split for PFIC-1 and PFIC-2, but given those numbers, I think that we'll see a similar kind of rollover in that. The open label extension study is important for us as a company because we want to learn about the long-term durability and sustainability of treatment with Odevixibat. Also, to kind of relate it to what Dr. Thompson says is, yes, we see these very nice bile acid reductions. What does that mean in the long term? In terms of the regulatory filing, both regulatory agencies have said it's going to be based on the strength of the PFIC-1 study.
Thank you, Pat. And are you able to say how many patients have completed 1 year or 2 year on the open label to an extent?
So we don't have those numbers available right now.
Okay. Thank you. And thank you.
Dr. Thompson?
Sorry,
Thank you.
What was the question?
It was about the 1-point drop, clinical
Oh,
meaning.
yeah, sorry, of course it was.
Drop, Dr. Thompson?
Yes, so absolutely. So in clinical practice, to be honest, most of us don't use scales like this. What we really quiz the patients about is how much the disease and the pruritus is affecting their quality of life. So I ask them questions about how much they're sleeping at night, how many times they're waking up at night, whether they're falling asleep at school, etc. So really the effect it's having on their life. Obviously, for a trial, you would need a more structured analysis. So what happened was that this scale was then used with families and correlated with those things that I think are clinically significant. And so the feedback was that on this scale, a 1-point change actually conveniently did correlate with those sort of clinical meaningful changes in life. So it was a separate correlation between the questionnaires about quality of life and those features and the scale.
Thanks, Dr. Thompson. And just Yasmin, to add in regards to PFIC2, we do have a fair number of patients that have been on therapy for well over a year, and we're looking forward to sharing more of that data in upcoming scientific meetings. Appreciate the kind words.
Thank you, guys.
Thank you. Our next questions come from the line of Brian Skorney with Baird. Please proceed with your questions.
Hey, good morning, guys. Congrats on the data. Great news for you guys, and great news for these kids as well. I guess 2 questions. First one, just wanted to clarify, when you say that both doses showed statistical significance for both endpoints, are you saying in sort of the primary analysis? I mean, obviously in the primary analysis, the combined dose groups are showing a statistically significant improvement compared to placebo. Are you also saying that each dose individually on a secondary analysis, or sort of low dose versus placebo, high dose versus placebo, in that analysis also showed both doses separately or statistically different from placebo?
So the answer is yes. So we did a hierarchical analysis of the endpoint. So the initial analysis was the pooled Odevixibat treatment groups versus placebo. And since we hit that primary endpoint, then we were able to step it down to high dose versus placebo and low dose versus placebo. And in each of those analyses for the 2 primary endpoints, overall was statistically significant compared to placebo, and each of the individual doses was statistically significant compared to placebo.
Great. Thank you for the clarification. And then, Dr. Thompson, sorry to kind of put you on the spot here, but maybe you could just kind of characterize, based on this data, what percentage of PFIC patients you think would merit use of this drug in, and maybe you could kind of say what would distinguish a patient you would put on Odevixibat versus one you would not.
So there was 20% of patients with PFIC type 2 are predicted genetically to have no functional protein and are very unlikely to respond, and we would not undertake surgical diversion anymore in those patients, and they were excluded from this study. So the inclusion criteria really would be 80% of type 2s and 100% of PFIC type 1s. And as I said to the previous questioner, I think because I believe that the side effect profile is sufficiently good, I think the threshold will be very low for trying it. And I think we need to drill down into the data and see exactly what it is. But on the bile acids, it looks like a third have had a really excellent response. We need to see whether in the details of exactly if we can predict which ones those are, etc. But in the absence of any further data, I think most people will be saying we will try the drug. And we will have to look at the data in more detail and see how long it is that we have to expose someone to the drug to decide whether they're a responder or not. But I think it's a matter of weeks. And so I think people will use the drug, and then obviously the responders will stay on drug. I think it will be very widely accepted.
Great. Thank you very much.
Thanks for the questions, Brian.
Thank you. Our next questions are from the line of Yoon Yang with Jefferies. Please proceed with your questions.
Thank you. Just want to, first of all, congratulations on clean positive data. I want to ask you what % of patients in the phase 3 had a prior PBD surgery and what you've seen in terms of response?
So, again, we've really just analyzed the primary endpoints in the overall population. We did enroll in PFIC-1 8 patients with biliary diversion surgery, and we're looking into the bile acid response and pruritus response in those patients.
Okay, thank you very much.
Thank you.
Thank you. Our next questions come from the line of Liana Masatos of Wedbush Securities. Please proceed with your questions.
Thank you for taking my questions and great data. So you mentioned that you're going to file for U.S. and EU regulatory review in early 2021, and you mentioned draft modules 1 through 5. What are the steps needed to have the regulatory documents ready, and what is the status of commercial manufacturing?
Yeah, thanks for the question, Liana. So from a regulatory perspective, as I said in my prepared remarks, we've actually been working on this for well over a year. We're literally dropping the data in now and making sure that it all ties together. Our guidance is that we will complete these regulatory filings no later than early next year. So obviously the team is working already feverishly to get these modules up and going and submitted in the U.S. and Europe. From a manufacturing perspective, I just will remind folks that, first of all, we used the planned commercial formulation in the phase 3 study. Second, that stability batches, registration batches, have been sitting there since last year, so we're in good shape in that perspective. And we have work to do, and that work is the work that we agreed with the FDA to be done, and that's being executed as we speak.
And do you have plans for a partner for rest of the world?
Yeah, Liana, the answer to that question is absolutely. As we've said many times, it is our intention to commercialize in the US and Europe, but we are looking for partners and have been, frankly, working with a number of potential partners in areas outside of the US and Europe over the last few months. So now that we have the data, we should be well positioned to move those discussions forward.
And you mentioned 10 reps. Is that for US and Europe or just US?
That's for the US and probably an equivalent number in Europe.
Thank you very much.
Thank you, Liana.
Thank you. Our next questions come from the line of Ed Arce of H.C. Wainwright. Please proceed with your questions.
Good morning, everyone. Thanks for taking my questions. And let me add my hearty congratulations on a very well designed and executed study and great results. Couple questions for me. First, with regards to the PFIC-2, the open label extension study, could you talk a little bit about perhaps Ron or Pat on the expectations that you may have for further serum bile acid reductions over the longer than 6 months of treatment, and how that could potentially affect payer discussions around any potential benefit on transplant, and what part of PFIC-2 would you be expecting to submit with the filing? And then I have a couple follow-ups.
Yeah, so Ed, as you can appreciate, the data has just come in, and we're in the process of analyzing a lot of this data. We will add into the filing whatever we have from a PFIC2 perspective. When it comes to access, we're really looking to put together a compelling access story, and as I said, the value document is already in place. We're dropping in the phase 3 data, and then we will be modeling out some of the long-term effects based on the data that we had, and that will take PFIC1 data, PFIC2 data, and NAPID data together to really make a compelling value story.
Okay. And then just one quick question for Professor Thompson. With regards to the rate of infections in these children, 52.4% versus 60% on placebo. Don't know if that's stat sig, but just wondering if you could discuss perhaps any thoughts around the sort of ancillary benefit of having the serum bile acid reductions and the reductions in pruritus, and what that might actually mean to the overall quality of life and health for these kids.
So, obviously pruritus is the major symptom, and that's the thing that really distresses the patients and the families. But as I'm sure you realize, it's a very difficult thing to measure, and we rely on these relatively simple scales. And a bit like pain, really, it's a perception. And in most of these young kids, of course, it's not even the perception, it's the secondary perception of the parents and what they perceive the child to be suffering or not. So it's absolutely no surprise to me that we see an improvement in pruritus in the placebo group, for instance, because this is an extremely miserable condition, and just being involved in a clinical trial changes your whole mindset. So, however, so I think it makes it pruritus as a difficult endpoint to achieve. And obviously I'm delighted that there is a clear statistical significant difference between the two groups, despite that inbuilt considerable hurdle. But as I say already, really, the differences that we've seen between the two groups are what look like clinically significant changes. And I haven't seen the patient level data either yet, but I strongly suspect that, like the bile acid endpoint data, I think we're going to see quite a dichotomous change. And I think many of these patients won't just have had an improvement in pruritus, I think they'll have had a very significant improvement in pruritus, and even many of them are hopefully a complete loss of pruritus. So, ultimately, I think that the question really is going to be not around the whole group statistical analysis. It's a question of how many patients really have had their whole disease transformed, not just improved, but really reversed.
And Ed, just to thank you for that, Dr. Thompson. Just to clarify, Ed, we had really good tolerability in the study. To your point about infections, that was the most common non-treatment related AE. And remember that this study, the age of the kids is around 4 years old. And those of you that have young ones, you realize that that's quite common for that. So just to provide a little clarity for that. Thank you for your questions, Ed.
Sorry, yeah.
That's great.
Thank you. Our next question has come from the line of Alan Carr with Needham & Company. Please proceed with your questions.
Hi, thanks for taking my questions. It sounds like you're still early in the data review process, but I thought I should ask these just in case. Any differences in response regionally or by age? And then also, can you remind me what the period was for assessments or interim assessments over the course of the trial, and did you see any trend over time in terms of a growing difference from placebo, or was this a quick effect that was steady? And then the other one around patient identification. Ron, you mentioned 8,000 to 10,000 PFIC patients. I'm kind of curious if you and maybe Dr. Thompson can comment on how many are known in the U.S. and Europe, identified and seeing a physician. Thanks.
Yeah, so I can take the first part. So in terms of the subgroup analysis by region, by age, by gender, all of those will follow on. Those are all part of the planned analysis, but we don't have that data yet. And similarly, throughout the study, these patients had visits every 4 weeks. So we will have data from the individual visits to plot out the time course and when we can start to see that separation between active and placebo. But these are, as you said, follow-on analysis that we'll be presenting later.
Yeah. So Alan, we estimate there's somewhere between 8,000 to 10,000 individuals in the U.S. and Europe that have PFIC. I think as you're aware, unfortunately, there is no published prevalence data or registry. So that is those are modeled numbers. We're really active now in careful work around the world, doing sort of site-by-site mapping to understand more about the patient opportunity, and we're going to be sharing that information with you as the year progresses. So thank you for your questions, Alan.
Yes, thanks.
I would just add
congratulations on the outcome.
I would just add, obviously, that genetic testing is now very widely available certainly in Europe and North America. And obviously that's the key basis to making the diagnosis. And that's become much more accessible in the last few years. So I think early accurate diagnosis is now widely available in those territories.
Great, thank you.
Thank you, Alan.
Thank you. Our next question has come from the line of Steve Seedhouse with Raymond James. Please proceed with your questions.
Well, congrats on the study success. Thanks for taking my questions. 3 quick clarification questions on the data, actually. The first one is about the placebo response in PFIC-1 patients. It just wasn't clear from slide 10 how the 5 PFIC-1 placebo patients did and if there was a placebo-adjusted drug effect in PFIC-1. My second question is on the GI treatment emergent adverse events, if they were distributed throughout the treatment period or mostly at initiation of treatment. And then my last question is just on the mean reduction data charts for the different metrics. Could you clarify what the error bars are there, if they're standard deviation or standard error of the mean? Thanks for taking the questions.
Okay, so in terms of the placebo response in PFIC-1, so we haven't split the placebo response out yet by the response within the groups PFIC-1 and PFIC-2. In terms of the GI adverse events, those were distributed throughout the time course. And typically those were very mild, moderate, and transient. Like I said before, we did have the one dropout of the active arm because of diarrhea, but for the majority, most of those were mild or moderate and in a relatively short duration. The last part was the mean reduction in the standard. So what's shown there is the standard error of the mean in the bars.
Thanks so much. Really appreciate it. Congrats.
Thank you, Steve. Thanks for joining us.
Thank you. Our next question has come from the line of Ritu Baral with Cowen. Please proceed with your questions.
Hi, guys. Thanks for taking the follow-up. I just wanted to ask Dr. Thompson and the broader team about the imbalance in background treatments on one of the slides and how you think that might impact the analysis of the various endpoints.
Well, and so this is Pat. So I don't think Dr. Thompson has the slides in front of him, but for UDCA, it was 90% in placebo group versus 76.2 in the Odevixibat group, and for rifampicin, it was 85% in the placebo group and 51% in the Odevixibat group.
So just from my point of view, and I can't add anything to that really, but apart from the fact that obviously we do use ursodeoxycholic acid very widely in these patients. I think if we're going to deplete patients of primary bile salts, probably replacing them with a small amount of ursodeoxycholic acid, which is much more hydrophilic and less irritating, is probably a good thing. So the combination is quite a logical thing to use Odevixibat in combination with that. But also rifampicin may be increasing some of the transporter expression in the liver. And so again, it may be something in which in combination is helpful. And obviously all we can say is actually, of course, there was greater exposure to both drugs in the placebo group. But I think in the future, combination, exactly understanding what combinations are perfect will be an important part of how we use the drug clinically. But I think we need to drill down into those data and see if there were any other differences we can find.
Yeah, I think, and we will. And I think just to remember in terms of study design, so patients had to be on stable drug therapy for UDC or rifampicin for, I believe it was 3 months prior to the start of the study, and those doses were to remain stable throughout the study. So even though these patients were on these other drugs, they still met the inclusion criteria of having significantly elevated bile acids above 100, and they had to have a pruritus score of at least 2 on the 0 to 4 point scale.
Yeah, thank you, Ritu. I think the background therapy is similar to what was in the phase 2 study. And so we're really pleased to have shown positive results on top of that background therapy. So thank you, Ritu.
Thank you. We have reached the end of our question and answer session. I will now turn the call back over to management for closing comments. Ron?
Okay, well, thank you all for attending today's conference call. It's always been our thesis that successfully treating cholestatic liver disease is about both consistent and deep reductions in serum bile acids. And today we back that up once again across multiple PFIC types. This is a transformative period for Albireo with a bright future ahead. Thanks for joining us today and for all of your continued support. Have a great day.
This does conclude today's conference. You may disconnect your lines at this time. Thank you for your participation and have a great day.